Devon Lukow from genetics will talk about cancers evolve drug resistance by shuffling their chromosomes.
So every day, scientists are working on developing new drugs to combat cancer and improve the lives of patients battling this disease. Recent advances have had remarkable results, putting tumors into remission and increasing patient life expectancy. However, for a large fraction of patients, this is only temporary, as eventually the treatments will become less effective as the cancers evolve to become resistant. This is because cancer is fundamentally an evolutionary disease. Kind of like bacteria, when cancer cells rapidly divide, they also start to diversify, acquiring new characteristics that make them more aggressive. And some of these characteristics include drug resistance. And that's why it's a key to understand how cancers can diversify in order to become or design better treatments that can provide better treatment outcomes for patients. So it's commonly thought that cancers acquire drug resistance through mutations or changes in the sequence of their DNA, causing a change in how a gene functions and thus providing resistance. And this has been linked to cases of resistance for lots of different drugs and cancer types. However, for a large fraction of cases of drug resistance, there are no mutations that are linked to it, suggesting that cancers have other ways that they can diversify to become resistant. And this is where my thesis research comes in. So one of the most common features of cancers is that they have an abnormal number of chromosomes. Chromosomes are the structures that contain all of the DNA in your cells. In cancers, as they divide, they'll often shuffle up their chromosome numbers, acquiring different gains and losses. So this is another way that cancers can become diverse, not by changing the sequence of their DNA, but by changing how much of it they have. So my thesis work was focused on seeing if we can shuffle up the chromosome numbers in cancer cells and if that will cause them to evolve more quickly. So to do this, I took cancer cells, I briefly treated them with a drug that caused them to shuffle up their chromosomes, and then I tracked how they responded to cancer treatments. And what I found was that while these cells that had shuffled chromosomes would initially respond to treatments, they would very quickly acquire drug resistance. But more strikingly, when I looked at the chromosome changes within all of these drug-resistant cells between replicates of the experiments, I found that the chromosome changes that they had were all the same, suggesting that it was these chromosomal changes that are actually driving the resistance. So in conclusion, my thesis research provides a new understanding about how cancers can diversify and acquire drug resistance, which may inform future treatment strategies that may target new vulnerabilities of these drug-resistant cells, or by designing strategies that can prevent these chromosomal changes from occurring in the first place and limiting the evolvability of cancer to acquire resistance in the first place. Thank you.